Hello guys, it's Adam the Science Tutor, and today we're going to be talking about how to make collagen fibers and what they are a bit, but mainly how to make them, and it's in about 7 steps. So what are collagen fibers? Collagen fibers are very important in the body. We use them for our bones, our skin, our tendons. We need this for a lot of different things. Our cartilage, a whole bunch of things. The main one we'll talk about is type 1. So there's about 15 types, but type 1 makes up about 90% of all collagen in the body, in the human body at least. And type 1 is responsible for your bones, your skin, and your tendons. Also for the cornea of your eye. So that's type 1. And to form this, we're going to have 2 alpha 1 chains and 1 alpha 2 chains. So 2 alpha 1 chains and 1 alpha 2. So that's 3 together, and that forms a triple helix. So it's in a triple helix. Collagen fibers are not sulfated, so they have no sulfate bonds. And collagen fibers are extremely stable, extremely stable. So cool. Let's talk about now the steps. We're about 7 steps in doing this. Ooh, another important thing about collagen fibers is that they're made up of 3 amino acids. You're gonna have proline, glycine, and lysine. What are good about these? Lysine and proline can both be hydroxylated. Proline is also a stabilizer, and glycine is just small, and it helps with the turns and the combination of the triple helix. So I'll leave this kind of up here, but proline, lysine, glycine make up the collagen fibers. So step 1 is gonna occur in the rough ER. So step 1 is happening in the rough ER. And we're going to have our messenger RNA, our ribosome, our polypeptide, and what's going to get this in here is the amine or signal peptide. That's going to get it into the lumen of rough ER. And we're going to create, so this creates a pro polypeptide. Cool, so step 1's pretty easy. We just created a pro polypeptide, and the signal peptide is what got our polypeptide into the lumen of the rough ER. So step 2 is going to occur in the rough ER, in the lumen of the rough ER. And what we're going to do here is just cleave that signal peptide off. It did its function. We're just going to cleave it off. So that broke. And that's going to create your pro alpha chain. So the next couple steps are going to be a little bit more confusing, but this is going to create your pro alpha chain, and the next steps are going to occur at the Golgi. And what happens at the Golgi? We're packaging things, and we're adding on to our polypeptide to add different functions. So we have our pro alpha chain right now. Step 3, it's going to happen at the Golgi. So step 3, we have our polypeptides. There's going to be a few of them in there. So what's going to happen in here is we're going to have this is our amine side and our carboxylic side. And these are all pro alpha chains. These are all in the formation of the collagen fibers. So what's going to happen in here is we're going to have a bit of more complicated, but we're going to have prolyl hydroxylase, and lysyl hydroxylase. And what do you think these are going to do? So our proline and our lysine are the ones that get hydroxylated. So prolyl hydroxylase will hydroxylate the prolines, and lysyl hydroxylase will hydroxylate the lysines. So we're gonna get, and it doesn't happen on all of them, but it does happen on quite a number of them. And depending on which collagen we're making, remember we have 15 types. Some will have more lysines hydroxylated, some will have more prolines hydroxylated. So we'll get these mix-ups, and this makes different alpha types. So we have these, and then we'll also have a lysine. Our lysine hydroxylases will also sometimes be glycosylated as well. So they'll be glycosylated in step 3 and hydroxylated to form our pre collagen chains, or pretty much our pre-alpha chains. So in step 4, however, one more key detail also, this needs absorbic acid. Absorbic acid. Common name for that is vitamin C. So where do we get our vitamin C? We get it from oranges. We get it. It's just a vitamin that we need to make our, or to hydroxylize our prolines and our lysines by our prolyl hydroxylase and our lysyl hydroxylase. So that's about done there. And then, so now we got our this is gonna be now step 4. I'm gonna reuse this picture. So now we have each of our alpha chains that are now hydroxylated, and so they're gonna do this number where we're gonna make our triple helix. God, you gotta excuse my poor thing, but it's supposed to be a triple helix. It's supposed to look like DNA. We're gonna have our carboxylic side over here, and our amine group on this side. Spontaneously, these carboxylic side will make disulfide bonds, and this will form your pro-collagen. So we're gonna make a pro-collagen. In medical terms or in biology terms, pro is like almost like pre. It's a step ahead of pre. So you have pre-pro, and now we have a pro-collagen. And spontaneously, our carboxylic sides are gonna form disulfide bonds. And I know what you're thinking when I told you collagen fibers don't have disulfide bonds. Well, we're going to talk about that in step 5. So we created our pro-collagen by taking 3 pro-alpha chains and combining them spontaneously with disulfide bonds. So this was step 4. Step 5. And this is all happening in the Golgi apparatus. So step 5. So we have our Golgi. Step 5, we have our pro-collagens. So we have a couple of these pro-collagens, and we're going to take 2 guys. We're going to have the N-pro-collagen peptidase and our C pro-collagen peptidase. And what's going to happen here is where both our N pro-collagen peptidase and our C pro-collagen peptidase are going to do is they're going to cleave both these sides off. So these were just formed making disulfide bonds. We're going to cleave this and cleave this side off. So it's going to cleave here, the N pro-collagen peptidase on the amine side because it has a nitrogen. And our C pro-collagen peptidase is going to cleave on this side. And we're going to create, and well, before we create this, we're going to have to get this out of the Golgi. Out of the Golgi and into our extracellular fluid, which is going to start step 6, but now that's going to create, once it gets into the extracellular fluid, you're going to create your collagen fibril. So not your fibers yet, it's going to create your collagen fibril. So I said we had 7 steps. We're going to create our collagen fibril. That was kind of step 6, is getting it outside of the extracellular fluid. It's not really step 6, it's still step 5. But that is what it is. Now we're going to talk about what happens when it gets into your extracellular fluid. So now that we're in our extracellular fluid, we're going to have our nice collagen fibrils. Let's see if I can draw this out a little better. Slightly. Alright, cool. So we have our collagen fibrils now, and they have been cleaved already. There's still going to be amines on this side, still going to be carboxylic acids on this side. It's in our extracellular fluid. What are we going to need to make this into a collagen fiber or into our collagen fibril completely? We're going to need these to be connected, and I know they don't have the disulfide bonds anymore. So I know you're thinking, well, how come they don't just peel or tear apart? Well, we're gonna have lysyl oxidase. Okay. I have lysyl oxidase. And what it's gonna do is gonna take it's on the lysine, either on the lysines, the free lysines, or the hydroxylysines, or the glycosylated lysines. It's going to oxidize them, and it's going to turn them into, so it's going to take the lysine or hydroxylysine, and it's going to turn this, into allylysine. Or hydroxy allylysine. So the lysine and hydroxylysine will turn into allylysine or hydroxyallylysine. This is happening from the lysyl oxidase. And so what's important about this step is that these are very reactive and they form covalent bonds quickly. So that's going to form your covalent bonds, keeping our structure together because we got rid of our disulfide bonds. Collagen fibers do not have collagen bonds. And this is going to complete the step of making collagen fibrils. So cool, now we have collagen fibril. Or at least collagen fibril is now finished, it's completed. It's now in the extracellular fluid. It's no longer in the Golgi. And so now how do we make collagen fibrils into collagen fibers? Well, these allylysines and hydroxyallylysines are very covalently bond, and so what happens is we have a bunch of collagen fibrils, and I'm going to draw them as blocks. So we have a bunch of these, that equals this, collagen fibrils, and what's going to happen is these allylysines and hydroxyallylysines are going to covalently bond to each other in a staggered way. So they're going to stagger on top of each other, and then that's going to make your collagen fiber. So this is your collagen fiber. And there you have it. You created your collagen fiber. This is now, that was step 6, and this is now step 7. So step 7 is the allylysine and the hydroxyallylysines are now going to covalently bond with other collagen fibrils and make the collagen fiber. And collagen fiber is extremely stable. It's not a lot of things can break it apart, and it has a long half-life. So as you'd imagine, your skin, your bones, they're going to be extremely stable. So you don't want them to have you don't want to replace them a lot. So they have a half-life of about a couple months. It creates a osteoclast, which is part of the bone, will create these things called collagenases. So you'll create collagenases, which will pretty much tear up the allylysines and hydroxyallylysines, creating collagen fibrils. Collagen fibrils will then be endocytosed and further broken down by lysozymes, and then we'll create pretty much the destruction of these. We'll take this into a lysozyme, and then lysozyme will create amino acids again, and then the process will repeat. So that's how we return back to there. But collagen fibers are extremely sturdy, extremely stable. It has great bonds. It's yeah, and it's essential for a lot of different processes and a lot of different parts of our body. So yeah, there you have it. That's the 7 steps of making collagen fibers. If you guys have any questions, please email me below, and yeah, ask away. Thank you guys.